I'm Dave Miklas, Executive Director of the DNA Learning Center at Cold Spring Harbor Laboratory. I'm here with Dr. James Watson, Chancellor Emeritus of Cold Spring Harbor Laboratory, and we're talking about sequencing the human genome and the FarGen Project. Thank you for coming, Jim.
I'm sorry I'm not with you, and I guess you could say the main reason is I'm 85 and I don't want to go from here to the Faroe Islands to Seattle to San Francisco and back here. There's a limit, I think, to my enthusiasm is greater than my physical strength now.
You were one of the founders of the Human Genome Project, and from your perspective, what has been surprising coming out of the human genome, and what have we found out?
I think, the overwhelming role of transposable elements in evolution, and they or their modifications of them comprise most of our sequence, and I don't think anyone would guess that and or studied it that way. I mean, that was a big surprise. It's now inconceivable to think of life without the knowledge. It's just very fascinating. Without the genome, we couldn't have done anything. So, the biology would have, in a sense, gone very slowly if there had been the Human Genome Project. It's created, an entirely new sort of branch of biology, genomics. And President Obama said, that was the best investment America ever made, was the Human Genome Project.
What is the big thing we still need to find out from the human genome?
How we develop, the basic, how do you go from a genome to a human being? And the big problem is sort of, how does development occur? And there's all sorts of, regulatory regions and DNA binding regions. So gene regulation is the sort of, in a broad sense, is the intellectually challenging field.
What are the big things that we need to figure out?
There was the big surprise when, we ended up with, 21 or 22,000 genes instead of the 100,000 that everyone was anticipating. But we can see there sort of exon regions, but we can't see the regulatory regions. And there's, been big projects begin to identify it, but we need to know how every gene is regulated. And when we started, we didn't know anything about microRNA, so the whole role of RNAs in controlling the final RNAs that we end up with, coding for proteins. It's going pretty fast, but I would say regulation is the big story. Yeah.
Now, you're one of the first persons to have your whole genome sequenced. What's it like to have your genome out there on the internet?
Well, it was very useful because it was compared by the people in Craig Venter's group, and his genome had been sequenced. So they compared us with regard to the polymorphisms we had in how we metabolize drugs. And, Venter is pretty normal, but I just don't metabolize beta blockers. And I don't metabolize antipsychotics. So if I would go, into a psychotic episode, they should very watch the dose. So, but I'm sure no one will be thinking about that if I do go into that. But I'm just saying that everyone should have that region looked at. So there are some regions where you want to look at everyone. And then there are other regions where it's not so relevant.
Now, there's a proposal to sequence the entire genomes of the whole population of the Faroe Islands. What's unique about that, and what might it tell us?
These people have been separated for about 1,000 years. So we're really looking at what can happen in 1,000 years. And, what are the selection pressures? And, have any new mutations arose and permit this, or is it just taking old ones and making best use of them? So the mitochondria, the women were largely from Scotland, or probably northern, that region, and the men were Vikings. So you have to know the Viking genome pretty well, and you better know the Scottish, and then see how it all works together. But if I lived there, I would say, it'd be fun, because someone's really gonna look at us. And it would be very surprising if some relevant information didn't pop up. Some allele which either, was good for you or bad for you and was more unique in the Faroe Islands than some other place. But I think it's a project which a large number of people outside the Faroe Islands would find very fascinating.
Now, as somebody with a lot of experience in this, what do you think will be the biggest hurdles that they'll find in setting up and designing a project like this?
I'm sure data collection. And, integrating the medical data with the genomic data. In England, you could sequence 150,000 or 250,000 people and try and integrate that with the medical records. And I'd rather, have a pilot project like the Faroe before I jump to the big English one. The group that are in Faroe Islands now is very bright people. Some of the cream of our scientists are interested in the project.
Jim, thanks for coming.